It is very clear that there is a global fertility problem.
You know, we all know that a semen analysis is a terrible test of fertility. Every time it doesn't work, patients sent back for a sperm problem. It's never an egg problem. It's always a sperm problem, but it drives me crazy because the man says to me, "Doc, what can I do now?"
What we actually found was not that the stem cells are not healthy. The defects are actually in the what we call the somatic niche, which are the cells that give the stem cells the instructions. Those are ones that are broken.
It's the toxic environment.
It's the toxic environment.
Yeah.
That's a really good way of putting it, Amy. So Paterna Biosciences has now succeeded in being able to grow at least sperm, whole sperm, in a dish from their starting cells.
It's almost science fiction, but it's science reality.
It's science fact.
I love polarizing topics, right? Because that's what actually changes medicine. You are actively changing medicine in the field of fertility that probably is not going to look the same 5-10 years from now.
We could solve not just the quantity problem, but the quality problem as well.
That is really exciting.
Yeah.
But what gets me up in the morning is the promise of this actually touching patients in as soon as possible.
Welcome to Fertility and Other F Words, the podcast that tackles fertility, sexual health, and everything in between. No topic too taboo and no question off limits. I'm Dr. Amy Pearlman, board-certified urologist with expertise in sexual, hormonal, and genitourinary health, and co-founder of Prime Institute in Miami, Florida.
And I'm Dr. Larry Lipshultz. I'm a board-certified urologist, specialist in men's health and fertility, professor of urology at Baylor College of Medicine, and director of our division of male reproductive medicine and surgery.
Now, Larry, you always have interesting things coming up in your clinic. So what was one of the interesting cases this week?
So this is a kind of a tragic one. So this gentleman is 51. He has a history pre-pubertally of bilateral undescended testes that were brought down, although his left is really high riding and has to kind of be dragged down to get a good feel of it. His wife is 33. She's a lawyer. She has PCOS, which is a problem with her ovaries, but not insurmountable. And they're trying to have a baby. He really wants to have a baby. I mean, this is very important for him. He wanted to have it earlier, but his wife is a lawyer, and she didn't want to have a baby at that point, so they've waited. He now comes in with an occasional sperm in the pellet. So to be a little bit more clear, a pellet, you take the sperm sample and put it in a centrifuge, which spins it around, and all those cells come down to a tiny little button. That button is known as the pellet, and it's taken out, and every part of that pellet is analyzed. And this gentleman had 6 sperm, non-moving, right? So we had to do something else. So he's slated to have...
Let me ask you a really quick question here. Does every infertility specialist centrifuge down the sample to a pellet?
They should, but I have no way to know. They should. I mean, that's kind of basic, right? So he's set up to have microscopic testicular sperm extraction. Sometimes we can get away with office testicular sperm extraction. And so we decide whether we can do that by doing a sperm aspiration. If we find sperm, then we just target him to be done in the office. If we don't, he has to go to the operating room. And we did it with this man, so he has to go to the operating room.
Okay, and just very briefly describe what's an aspiration.
So we use an 18-gauge needle. We give the patient a cord block, and then we use the syringes that are used for breast biopsies. I don't know where they have the little thumb thing for you. Put your thumb in and you aspirate. And then we go in and out of the testis, not coming out, but within the testis 10 times. At the last time, we put a lot of suction, gently extract it. As it comes out, it will usually pull a tubule, and another doctor with us will reach in, cut the tubule off. It goes on a slide. We tease it. We'll sometimes send part of it to pathology, and then we check it for sperm.
And that can be done in the office, very well tolerated.
Oh, yeah. The cord blocks are amazing.
Yeah.
We're using cord blocks now for our vasectomies.
Yeah.
And they're great. Anyway, so on this man, no sperm when we did that procedure, so he has to go to the operating room. So the day his wife's eggs are coming out, we take him to the operating room. You can sometimes do it the day before and incubate overnight, but I believe this was the same day. We go to the operating room, and we do a microscopic testicular sperm extraction. Now, you know what it is. I don't know how many of the people out there have any idea what it is, but if you can imagine taking an apple, slicing it in half, opening it up, or an oyster shell even, and that's how we do it with the testicle. It's really a very invasive procedure.
Yeah, so you open up, you cut the testicle in half, you open it up, and you kind of comb through the testicle.
Looking for tubule under the microscope. Now, of course, the testicle is not in half because we keep the back wall, so it is basically an oyster, right? Okay, so in this man, and what we do is we check in the operating room to see if there's sperm, right? So I go back in the notes, and I remember this case. At the time, we found sperm. So we have sperm in one side at the bottom, the other side at the top. We label that on the tubes. So when it goes to the IVF lab, they know these are the places where we know there's sperm, right? So apparently they found enough sperm. She had 16 eggs. So nice number of eggs, right? Mature eggs. And apparently they had enough sperm because they did ICSI, intracytoplasmic sperm injection. That's where you put one sperm in each egg. And they had enough to do that. And they wait, So the next day, high fertilization rate. For blastocysts, 5 days later, they have 3 blastocysts. 2 of them are aneuploid. 1 is healthy. They transplant it back in. It doesn't survive. The couple are distraught, right? They went through all this, all the lead up with the injections for the woman and the man who desperately wants the child, right? So I go on and I look at the notes, and they say, we feel it's a sperm problem, and they send the patient back to me. There's no reason to think it's a sperm problem. I mean, we know from data that we'll talk at a later meeting that the testicular sperm will often do better than the sperm that are there, we call cryptospermic, very low numbers, right? So the man's distraught. My feeling is, having done this a long time, every time it doesn't work, patient's sent back for a sperm problem.
Yeah.
It's never an egg problem. It's never somebody slamming the door on the incubator. It's never a new technician. It's always a sperm problem. And it really is so frustrating to me. I don't know whether you've been in this situation, but it drives me crazy because the man says to me, Doc, what can I do now?
Yeah.
I mean, another repeat microscopic testicular sperm extraction?
Yeah.
Very difficult and not that likely to work.
Yeah.
I mean, we're probably going to do it because I know where the sperm were, but should they change IVF doctors? I mean, what would you advise?
Well, I think that's a really interesting topic and very relevant to today's discussion.
Yes.
Because we're talking about when we look at, even just like a semen analysis, which is such a crude way, but it's like one of the only tools we have to assess, the health of the sperm is like, I guess we don't know yet if it's a sperm problem. It could be, but we need better diagnostic tools.
Well, he did have whole genome sequencing. We didn't find anything.
Okay.
So it's not something that he's putting into the sperm through a general genetic abnormality.
But wouldn't you say that men who have fewer sperm found either in the ejaculate or in their testicle with the procedure that you did, isn't their quality of sperm different than someone who has a normal count?
Yes, but the person with a normal count wouldn't be having this procedure, ICSI.
Yeah.
Right, they wouldn't be doing IVF.
Yeah.
So the question is, are your outcomes worse than a patient who has ejaculated sperm? Not that statistically significant.
Yeah.
I mean, the fact that they only fertilized 3 out of the 16 is a problem. And then when you go back and you look at the IVF data sheet, which I did, it says eggs look granular with debris in the perivitellin space.
Yeah.
I mean, I don't know, I've got to call them up and say, is this really bad? Is this, But the point is, what bothers me is telling the couple that it's a sperm problem, putting the onus on me now to do something different, and I don't have anything different to do.
Yeah, that's challenging. Yeah, I don't have any similar examples.
Now, if I had it, as I've told you before, if I had this to do again, if I had my career to do again, I would have my own IVF program. I would get somebody who I like, a really personable, talented reproductive endocrinologist, and we'd work together.
Yeah.
Because then it couldn't be you, me, whose fault is it? But it's a blame issue, and it makes me very uncomfortable at this point.
You know what's very interesting in what you just said is your relationship, I think, with some of your colleagues is very similar to some of the discussions or non-discussions that couples have together.
Yeah, I was thinking of the same thing, the blame issue, right?
Where it's either, it's his problem, it's her problem, when the reality is oftentimes it's a problem or an issue with the man and the woman.
Right.
And perhaps in this case, it's an issue with both the egg and the sperm.
Exactly.
Which is probably the more likely,
So, there was a
here.
time at Baylor, because we've been through 3 iterations of IVF programs. In the initial one, we used to have a meeting once a month when all the couples in cycle were discussed. It was very educational. My fellows got a lot out of it. They learned a lot. But we had a chance to brainstorm together. And now, since IVF has become so busy, it's kind of like off the table.
Yeah. Well, that certainly seems like it would be an easier solution than you starting your own IVF program.
Well, I'm not starting it now, but you can start it. I think you should start it.
So I want to tell you about a patient that I recently saw, and I have a practice in Miami, Florida. I share that practice with my twin sister, so we see a lot of mutual patients. So this actually talks about the
she's a
collaborative.
gastroenterologist.
She's a gastroenterologist. So this is on kind of like that same vein of like, how should we be collaborating with some of our colleagues?
Right.
And so, I mean, I'm very lucky. Her office is literally 3 doors away from mine, so she can just like knock on my door and be like, hey, I'm seeing so-and-so today. I just wanted to give you an update. Like, it honestly is that simple in our practice. So we see a 47-year-old male, and he had come initially to see my sister because he is overweight and wanted to lose weight. And he especially had a lot of fat around the midsection, which we know that fat around the midsection called visceral adiposity is a huge risk factor when it comes to cardiometabolic disease. And it correlates with the fat around the organs, which is especially dangerous. So this guy was started on a GLP-1 medication. Okay. So what is in all like these media headlines is how people are losing an incredible amount of muscle when they're losing weight, right? We see the hashtag Ozempic face, Ozempic butt, right? But the medications, they don't actually cause someone to lose muscle. It's a fact that a lot of people, as they lose weight with whatever means it is, are not getting enough protein, are not actually doing resistance training to stimulate the muscles, and their hormones aren't optimized.
So wait a second. So
Yeah.
if my patient
Yeah.
is going to take a GLP-1 or one of those
Yeah.
derivatives, and they do not want to lose muscle, if they continue or increase their resistance training, eat the prescribed amount of protein, they're not going to lose muscle?
And you have to optimize their hormones. You cannot have 2 out of 3.
They are optimized because they're my patients, and
So,
that's why they came.
yeah.
They won't lose muscle.
So that's what we're finding. And we haven't published on our clinical data yet.
But you should, because it's very important.
It is very important. It's super fascinating. And it's interesting because a lot of these pharma companies now are looking at different novel therapeutics to mitigate muscle loss. Folks, we already have the solution for people who are candidates for hormone therapy and have lower levels. So in this guy, he's 47, in the process of his weight loss journey. He's lost 40 pounds. Guess how many of those pounds are muscle? Just give me a number.
5.
1.
Oh.
1 pound of muscle.
Well, that's really unusual.
That is, but it is usual in patients when we do those, when we optimize those 3 factors,
Right.
okay? His body fat has decreased by 11%, 39 to 28%. And mind you, we are tracking this with a bioimpedance scale because you cannot track what we do not measure. So these patients of my sisters get on this scale every single month. We have this incredible data.
But that's such important information to get out there.
Incredibly important information. And importantly as well, he's lost 8 inches around his midsection.
Wow.
8 inches.
But he is going to end up with this big hunk of loose flesh.
Well, some people do, but I saw him the other
I'm not
day.
saying that's not a sign that nothing's working.
Right.
It's a sign that everything's working.
Well, that's some of the concerns in patients is they don't want to lose all the weight they might need to because they are worried about loose skin. And I don't know if that's a concern of his. I mean, when I recently saw him in the office, like he looks
How
great.
old is
He
he?
looks healthy. He's 47.
Is he still interested in fertility?
He's not interested in fertility. Yeah. And I would ask all these patients, what their interest is. But you can't have 2 of the 3. And what I find especially interesting
2
in
of
this,
the 3 meaning
2 of the 3, the protein,
Right.
resistance training, and hormone optimization, right? And what I find really
Can't you
interesting.
make like a thing out of like with the first letter of each one? So
Probably.
some type of a reboot with.
Yeah.
We'll have to think about
Yeah,
that.
we'll have to put our heads together. But sometimes we look for these very sophisticated ways of treating patients, and sometimes we have to look. The more innovative therapy is to just look at what we already know in the basics.
go back to the basics.
Go back to the basics, and that's what we found with this patient and a lot of other patients. And mind you, this is not just in men. This is in women too, right? You get a perimenopausal or menopausal woman, put her on hormone therapy. Same thing: resistance training, protein intake, and you're sparing muscle as well.
That's really important.
Yeah, I love it. So let's talk about some breaking bedside news here.
Okay.
You have a fun story.
I do. So this is from Men's Health.
Yeah.
Right? My new go-to
Your
now
go-to.
for breaking news. And I want to read you the beginning of this article.
Okay, I'm ready, Larry.
It says, there are many things one can do to relax at the end of the week. Dinner and drinks with your partner, a baseball game, the movies. I chose to spend the last Friday night in April inside a downtown Los Angeles soundstage designed to resemble a Vegas title fight arena to watch a sperm race. Seriously. They had these guys dressed like contenders. They would come up to the microphone, the champ, the challenger.
Yeah.
And they did a sperm race.
Yeah.
They actually put the semen in a specially designed chamber under a microscope. The mic was projected up on a screen, and you could watch the sperm moving.
Yeah, and they did a race.
And they raced. And there was a winner and a loser. And the sperm champion.
Yeah. Okay. So
it was amazing.
Let's go back to couldn't this actually
love
happen?
this,
I know.
though.
I love this too. It's definitely.
But let me tell you something.
Yeah.
The premise to do this, the reason to do this, by this fellow named Eric Zhu, was to raise awareness for infertility. and decreasing sperm counts. I thought that was a great way to raise awareness. I mean,
think
you know?
yeah, it's very clever.
I liked it.
Yeah. So, okay, let's just talk about this from like, can this actually happen? So when you're looking under a microscope or through a microscope
Right.
and you're looking at the sperm, what are you seeing? Describe for the viewers.
I see sperm moving in all different directions, around in circles,
Yeah.
going this way, going that way.
They're lost a little bit.
Well, they can't find that egg.
Yeah. So is it possible, like, could this really happen that if you put sperm in this specially designed sort of chamber, like a racetrack,
Right.
would they go to the finish line? How do they know where to go?
Well, I don't think they had much choice because
Yeah.
it was a very narrow chamber.
Okay.
And don't forget, sperm are deposited in the cervix. They find their way into the uterus and they go up the fallopian tubes.
Okay.
They don't come back out of the
Okay,
cervix.
so they're not directionally challenged.
No, they're smart.
So maybe because you see a lot of these patients coming in and, a lot of people are giving, sperm samples, maybe you'll be the next host of a race like this. Maybe
We
there's some
are.
collaboration
Yes,
opportunity.
at Reliance Stadium. I think it seats 70,000, right?
All right.
We're going to do it.
Hey, don't forget the little people when you put on that event. I want to
I
be
know.
there, front
You'll
row
be
center.
there. You'll be there. So thumbs up or thumbs down?
I'd give it 2 thumbs up for
I'm
sure.
going to give it a thumbs up. Although, do we know it's true? We're going to look into
We'll
it
look
first.
into it.
I want to try to have this gentleman, Eric Zhao, on
Yeah.
the show.
All right, we'll see what we can do.
Okay.
Welcome to today's episode. I am so thrilled to introduce our remarkable guest today, Dr. Alex Pastuszak.
Alex is a urologist, and more than a urologist, which in itself I think is pretty good. He is a scientist and a serial entrepreneur, currently serving as co-founder and CEO of Paterna Biosciences. He holds a Bachelor of Science from Yale and an MD PhD from University of California, San Francisco. He completed his urology and male reproductive medicine training at Baylor College of Medicine. That is where we initially met and have remained good friends ever since.
With over 15 years in healthcare and biotechnology, Dr. Pastuszak has led multiple startups, including Vault Health, and is an associate professor of surgery at the University of Utah, where his research focuses on men's health and reproductive health. Today, he's here to discuss groundbreaking innovations in male infertility.
So Alex and I go back a long way.
Definitely a long way.
Yes. So we met when you were an early resident in urology, Baylor College of Medicine, where I was and still am, unfortunately. No, I'm serious.
The world is a better place because you are.
I know.
And have been.
Anyway, and we've known each other since. And
That's
have
right.
stayed in close contact since, as I've watched your meteoric rise from a country doctor to a CEO of a very exciting organization. So going back to the days when you were just a doctor, when did you first start thinking about the entrepreneurial, scientific / industry type situation?
Yeah, I really appreciate that question in the sense that I still ask myself that same question.
Why or when?
Well, both. Because the job I do now is very, very different than just a job at an academic healthcare center.
Right.
But to get to your question, so look, I think going back, my life, one of my life's goals has been to have positive impact on people, right? Less so on people's bottom lines or whatnot, but really on just the health of people. And so when I was growing up, I knew I wanted to be a doctor when I was 6, which fit really well with my mom's understanding of what my life would be because she wanted me to do the same. But, when I started getting into research, some of my mentors said, hey, why don't you think about doing an MD PhD? And I said, well, why would I do that? And they're like, well, you could do the research part too. And I'm like, well, that's interesting because it lets you think differently. And then as I was finishing my MD PhD, I was thinking about, well, how can I go out there and have an impact, right? I could go the academic route and do the research and, figure out the cures and take that pathway, or, potentially I could go onto the industry side. And I actually almost left medical school and became a venture capitalist, right? But my mentors at that time said, well, don't, you are already accepted to residency and you should finish because literally they said, you will be worth more at the end, right? And honestly, that didn't come back that didn't track in my mind until about 2 years ago, right? When I actually realized that they were right. But to again, finish up on this, the company side was a bug that I think I've just had in me for a long time. As you know, I started my first company when I was still a resident, which I wouldn't recommend to anybody. That was like working 40 hours a day. And ever since then, there's been this drive to do more, to do better, but really in the vein of helping people. And I think one of the challenges that we have, and it's growing, especially at the academic healthcare level, is I feel it's getting harder, or it has gotten harder, hopefully it'll get easier, to innovate in that space. And so I made a conscious decision to step out.
Right.
Because it's much easier for me to work with a small team and have a group of invested investors and have the money and the flexibility and really I hate the word power, but also the power to just come out and make quick decisions that drive outcomes that then impact people.
And Alex, you and I go way back too, because
We do.
when you were a second-year resident, I started doing research with you, and you taught me how to be a researcher. And I remember
What was your research project?
It was actually...
Well, wasn't it it was one of our projects,
It was
at least.
a project that has changed the trajectory of my entire career. And it was on guys who had a history of prostate cancer, who had their prostates removed, or who had radiation therapy, who were put on testosterone.
Yes.
And I was tasked.
Important study.
Yeah, a very important study that laid this groundwork on top of some of the other colleagues that we work with, like Abe Morgenthaler. And I remember I would go in on the weekends and after hours, and I would look through all the paper charts when they had paper charts, and I was looking for all the PSAs, every single PSA could find. I was on a manhunt for them,
Right.
And ultimately, we wanted to know, do the PSAs go up, or is there a higher risk of biochemical recurrence in guys who had a history of prostate cancer? And we found that there wasn't. And
Which
that
changed a lot.
It changed, yeah. And that was, I learned how to be a researcher. I learned not to take offense when someone looked at my manuscript and had red everywhere, because I realized that for someone to sit down and read your work and put red everywhere, it takes an incredible amount of time and effort. So now I appreciate the red on the black, right? And I learned that to do research in a polarizing field is where the fun is at. I love polarizing topics, right? Because that's what actually changes medicine. And that's going to be the focus of a lot of today's discussion. You are actively changing medicine in the field of fertility that probably is not going to look the same 5-10 years from now.
Well, that's the hope, Amy. And I like your point about polarizing, because even now there's a polarizing discussion about the work that we do in terms of potential impact and the sort of, I won't call it a Sisyphean task, but the significant task of, not just getting your hypothesis out into the public space, but also proving it. So looking forward to the discussion.
Yeah.
So walk us through. So, you got an interest in research, an interest in entrepreneurial pursuits, but you were still a resident.
Yeah.
So then what happened?
So it's, there's been a consistency and also a stop-start nature to the last 12 years. The consistency was that there was always some sort of company that I had either been a co-founder or founding executive of, or been involved with ever since, that was usually on the small side. So just to tell the story quickly, after ConsultLink / Health, which was a digital health startup, really taught me how the healthcare system works and how difficult it can be for small companies to have an impact, even though they might have a great product.
Right.
After that really came some of the work that I was doing with Allotrope Medical as as an advisor, and that was Albert Wong's company. He sold it a few years ago. And then when I left Houston and went to Utah, I was asked to join the founding team of Vault Health, right? Which at its inception was a men's health focused sort of health tech-enabled healthcare services company, right? Where the goal there was to give men care via telehealth and at-home visits, right? So it was changing the paradigm of what clinical medicine typically was. And this was back in 2019. And, what was unique about it was, the team and I had figured out how to, create a nationwide practice, which we actually grew to over 1,500 practitioners at one point, and to get men hormone therapy as well as other therapies, right? We weren't just, we weren't going to go down the line of all we want to do is give men testosterone, right? You and I have never believed that. We've always believed in actually taking care of the patient, right? But we were also expansive in terms of covering ED and some elements of other aspects of men's health that could be managed via telehealth and kind of grow beyond that, right? Fast forward into the beginning of 2020, we hit the COVID pandemic and it hit us smack in the face. But we were a really smart and agile team. And we pivoted very quickly into COVID testing. And it was literally when we talk amongst the team about that, it was capturing lightning in a bottle, because we were fortunate to have a great academic partner, Rutgers, who had developed a saliva-based test, which already had an EUA from the FDA and was ready to go before day one. So we're talking February of 2020, moving into March, when we're having the discussion over the course of 8 hours overnight of, do we flip this company into a COVID testing company? And by 5 a.m. the next day, the answer was yes. And within 7 days, we had built all the software infrastructure and pivoted the company to be a COVID testing company. Right?
So you made lemonade out of lemons.
We made.
I didn't make that up.
What is the alcoholic version of lemonade? I don't know. Anyway, that's what we made, and that thrust us into the national spotlight. It obviously helped us do quite well, but we also did over 14 million COVID tests over the course of 2 years. And we were one of the biggest COVID testing providers in the US. At the same time, we also were building other functions that were focused on diagnostics. We built a diagnostics platform that transcended just COVID testing, that had to do with blood testing where patients could acquire the sample at home. We used new methods that were painless acquisition of blood, that were not like these paper-based methods that don't work very well. And, we were really pushing the edge of how healthcare can be administered in the home. And we built a clinical research platform as well, because halfway through the pandemic, not even halfway, we had AstraZeneca and a couple of other large pharmas who were doing their phase 3 COVID trials, knocking on our doors and saying, hey, you guys have this great at-home COVID test. Can you help us with our clinical trials? To which we said, yes, of course. But that was another sort of foundational business unit. So anyway, the bottom line is, amazing experience. Did a lot of good for many people, and and then we've exited that company over the last couple of years. The board said, let's print the win, and then after that came.
And really quick,
Paterna.
and you were on the phone with Elon Musk at one point during your Volt experience. What's it like talking on the phone with Elon Musk?
So for all the criticism that's been hurled at him over the past months, that Elon really knows what he's talking about, and it's kind of amazing to sort of watch from the sideline, but then to also talk to the man, right? And Amy, you and I were talking before we started this. What was the topic of discussion? We were actually talking sort of the biophysics of qPCR and what how the tests that we had developed worked and how it compared to other tests, and really getting down into the nitty gritty. And he understood it all, right? It was like talking to somebody who'd been in the field for a decade or 2 decades or their entire career, So really exciting, kind of fun. Bummer had to wait 10 minutes for him to get off his plane.
So let's talk about what you're currently working on. And, a lot of people that are going to be watching and listening to this podcast are kind of like the typical people walking around, and they're not these very, high-level scientists. So talk to us about the very basics. Why is there a need in treating men with fertility?
And also how you got into this company.
Yeah. So, well, let's sort of take a big step back and talk about the field of fertility, if you will. I think the three of us know a little bit about it. And then talk about the need, and then talk about how this all came to pass. So look, from a fertility standpoint, number one, we've all been in this space for a long time. You arguably started the field,
well,
and that there's been a problem for a long time, in the sense that when you think about the emphasis on care over the last 4 or 5 decades, it's been on women. Right. And part of that, and look, I'd love to hear your opinion on it, but part of that has been because the big breakthroughs have been in women, right? Like the inception of IVF in 1978, the inception of intracytoplasmic sperm injection, where you take one sperm and put it in one egg, 1-to-1 ratio in 1992, right? Those were big milestones in the fertility space, right? But that's where the field was and has been, right? It's been a small group of people, such as Larry, to an extent myself, who have really sort of underpinned the emphasis and the attempt at treating men, right? But we're not doing a great job, right? When you look at, I know that when I see my male patients, apart from what we can do today, which is some medications, a couple of surgical procedures, we kind of fairly quickly get to a point where we have to say there's not much more we can do, right? And that's not really acceptable. So,
but I think part of that problem, and you have not gone there yet, is the limitation of the diagnostic space. Because we all know that a semen analysis is a terrible test of fertility. Yeah. And now I think we're starting to see some innovative testing, but if you were to go in another direction at some time, that's where you got to go, because we can't treat people that we can't diagnose. And that's one of our big dilemmas.
Yeah. And I think drilling down further on that one point, the fundamental problem is you can't select a sperm and figure out right now if that sperm is the right sperm without destroying it, right? And by right sperm, we know that from a fertility standpoint, the genetic and epigenetic package, so what's in the genome, the DNA of that sperm, is really what's important, right? Not what it looks like, whether it twitches, or whether it's got, 3 fingers, right? And we can't interrogate that. I think taking another even bigger step back is looking at fertility rates across humanity, right? We've got a good enough signal now that independent of whether you and I or us think that semen parameters are actually getting worse and sperm counts are declining, right? It is very clear that there is a global fertility problem, right? We are on a trajectory since 1950 at this point, or 70, where the birth rate is steadily declining to the point where in 15 years, we're going to be at a point where humanity cannot reconstitute its population at its current level with just the technology we have today and people, right? And whether that's social, environmental, biological, a combination of all of the above, it's a problem, right? And so we need to bring technology to the fore in order for us to solve that problem, right? So that's a big part of what we're working on at Paterna. So Paterna Biosciences is a company that was that hired its first employee in 2023 and is focused and has now succeeded in being able to grow at least sperm, whole sperm, in a dish from their starting cells.
And could you tell the viewers why that's important? I mean, who would that address, and how common is that?
So, when we break it down simply, there are 2 problems in the male fertility space. And we can talk about females as well, because we're working on that too. But in the male fertility space, there's a sperm quantity issue. So there's about 20% of infertile men who don't have sperm in their ejaculate, right? And then there's a quality issue, right? So, there are about 200 million men around the planet who have poor sperm quality. And regardless of what you do, those men will not be able to get their partners pregnant. And their sperm, when used in IVF, will also have a low or no success rate, right? So, we're looking at helping these at least 250 million men around the planet that have these problems. And, when we think about the couple, when we think about people wanting to have families, we talk about IVF very glibly. Like, we talk about it as in, there's this great technology that's going to help you have children. The truth of the matter is it's not that simple, right? Many people, especially women once they hit their 30s and guys as they get older, it's not one and done. You're not going to have one IVF cycle to get pregnant. You're going to need 3, 4, 5, 6. And when you get into your 40s, 8 plus. And that is really taxing on the patients, on the couple, right? I mean, it is very taxing. It's incredibly taxing on the woman, because they go through weeks of drugs to stimulate their ovaries to make eggs. Then those eggs have to be retrieved. Then they need to be fertilized. Then they need to be tested. And this is all very wearying, right? So if we could improve the success rates of IVF, if we could bring in technologies that will reduce the burden both financially, emotionally, and physically on these couples, then that's going to be a huge win, not just for everybody involved, but for all these patients.
I have a very important question to ask you, just in your personal view. Do you think that the exponential increase in IVF is biology-driven or venture capital-driven, business-driven?
Great question. I think it's a combination, but I do think there is a biological factor. I do think that, especially...
given that there is a biological factor, what is the impact of all the money going into it in terms of the exponential growth? Do you think it's for patient care, or is it for profit?
I think it's both. I think it's more. Look, I mean, I'll say I don't necessarily trust private equity to do healthcare, right? And I know I've talked to friends who are in IVF groups that are PE owned,
right?
And the focus is on the bottom line, for sure. So I do think there's a driver there. I do think, though, that beyond that, there is a true problem with fertility, and it's not going to get any better.
Right.
Larry, what do you think about the patient versus the profit with IVF facilities?
I mean, I don't want to make a lot of enemies, but I see it every day. I just see couples, 6 cycles, 4 cycles. I saw a couple, unbelievable, that they were going to, and I don't know whether it was a Mexico-related thing where the patients here in Houston, the woman was triggered. Well, not triggered. She was treated. She made multiple eggs. The day before she was going to be triggered to release the eggs, they go to Mexico and do the procedure in Mexico, right? They did it 4 times. I then see the man, which is so common, multiple failed cycles, then I get the male patient. I see the male patient, I do a semen analysis, his sperm are all dead. He has no moving sperm. And I tell him that, and it's kind of, I said, doesn't this upset you? You've gone 4 times to Mexico. Didn't they ever tell you this? And I was much more upset than man was. He just didn't, it didn't register that he had been taken advantage of. And I just see that in less significant ways repeatedly. And I just worry about what's happening to healthcare in the space of infertility. It's really because these couples are so driven, as you well know, and they will do anything to have that baby, and they become ripe to be taken advantage of. And that's my whole. And certainly there are great IVF programs. There are great reproductive endocrinologists who we refer to as REIs. They're great, but as a whole universe of treatment, I just think it's ripe for people coming in and taking advantage of these couples.
And just to ask a follow-up question, is the problem that the men are not getting properly evaluated?
That's a huge part of the problem. It's not the problem.
Right. Yeah.
But I saw a couple this week, very well-educated couple. They had gone through 4 cycles, and and somehow they got to me. And she said, I can't believe that this is the first time we're hearing that the male should be evaluated. Come on.
So
that should not happen.
No, absolutely not. And I want to touch on the point that you made that a semen analysis is a terrible way of looking at fertility, and yet that's the tool that we're currently using. So what are the ways in which that's a bad tool, but what are the ways that it can be helpful? Because it does drive a lot of your practice management.
Look, we know you need sperm, and we need to have to be moving. So those 2 things are very easy to determine. The problem, as Alex was getting to, is you can't tell by looking at them, are they healthy sperm, right? And I think we're starting to see some new tests to address that issue. But right now, the semen analysis is just a low bar to get over before you get put into another type of treatment. So let's get back to your story.
Yeah, sure. So there's the infertility issue. There's the global fertility decline, right? So and look, I'm not an alarmist, right? That is not how I like to live life. But what I see in the data is not comforting, right? We are, and part of my mission, and as a result, my team's mission, is to bring new technologies to bear that help us help more people have healthy babies, but also help the human population, right? There is a part of my mission, my own internal core, that wants us to bring these 2, and we'll talk about the sperm part, but we are also focused on the female and making amazing progress in that space. Bring these 2 in vitro gametogenesis and gamete maturation technologies to bear, along with new emerging technologies, like we'll call it the artificial womb for now, but there are some really exciting companies out there that are building technology that lets you take an embryo and actually gestate it outside the human body.
Wow.
And that sort of trifecta of technologies, I think will, in the next 10 to 20 years, really kind of more durably solve the problem of infertility, along with everything that you've been saying in terms of diagnostics, right? We have crap diagnostics. And coming back to, where this all started, it was asking the question of what could we do in specifically the male space, right? Because all the founders are focused on the male space, that would move the needle more than, say, new technology to figure out how to select a better sperm, by looking at its shape and movement, right? Like you guys have probably seen some of the AI-based technologies, which, they may incrementally help, but they're not going to close this 25% of IVF cycles fail because of a male factor delta, right? 50% IVF failure rate, 25% of those 50% that fail are because of a sperm problem-ish. Like, these technologies will not close that, right? So we asked the question, what could we do? And when we sort of walked that back, the answer was, well, if we could figure out how to grow sperm in vitro from the natural starting point in the testicle, it's called the spermatogonial stem cell, but let's just call it like the upstream cell, and be able to oversee and quality control that process, then we could solve not just the quantity problem, but the quality problem as well, right? And we didn't necessarily frame it in our minds at that point. Like, really what Brad Cairns, who's the scientific founder of the company, did was he asked the question, well, what does normal look like? And by normal, we mean what is the normal programming for spermatogenesis? And so about a decade ago or so, there were a couple of new technologies that were coming online. One of them was called single cell RNA sequencing. And the power of that was being able to take a single cell, take the basically the transcriptome of that. So those are all the instructions that are kind of telling the cell what to do at that point in time. And to be able to sequence those instructions and take a snapshot of that cell at that period in time. And then string that together using computational biology to create a map of what all the cells in the testicle are and how they are talking to each other. And that was really key to giving us the roadmap for what we've done since then. And essentially what we did with that information is we use that information to figure out what all the chemicals are that tell the cells to go from stem cell to spermatogonium to round spermatid to sperm, right? Those are all subtypes of a sperm, right? And we recapitulated that process in vitro. And that took us about 2.5 years to do, right? And that's what's really led us to get to this point. But at the same time, we are able to, because these cells are not growing in the seminiferous tubule, right? In the context of all the other cells, we're able to just watch them. We're able to kind of pluck out a few at a time. We're able to figure out ones that have genetic and other types of defects. We can throw those away, and we can keep the ones growing that are going to be good and healthy. And those are the ones that we get to sperm. So anyway, I've said a lot.
Wow.
No, but I mean, it's almost science fiction, but it's science reality.
It's science fact.
But the thing that concerns me, and correct me if I'm wrong, is that the problem we have clinically with male infertility in this space is that we're starting with with baby cells that are not healthy. Hence, they will produce or not produce a sperm cell, and if they do, it won't be healthy.
Yeah.
And so the challenge is to take stem cells from someone who is not healthy and make them mature into healthy sperm.
So
and is that what you're doing? What is the source of your stem cells?
So it is from the biological or the would-be biological father. And let me actually correct a wrong around that, right? So that's what everybody thought until we did the work that we did, right? So what we actually found was not that the stem cells are not healthy, because by and large, there are actually very few defects in the stem cell that don't allow us to get to a healthy sperm. Right? The defects are actually in the what we call the somatic niche, which are the cells that give the stem cells the instructions. Those are the ones that are broken. The stem cells themselves, they're chilling, man. I mean, those things are like little kids playing on a playground. They're just fat and healthy for the most part, and they're just going to do their thing. No, and I say that glibly in the sense that was a big revelation for us for two reasons. Number one, even guys who don't make sperm have these stem cells.
Right.
Right. The only question would be like Sertoli cell only, but we're not going to get into that today. Right. And as a result, unless they have a genetic defect in a very specific cellular process, in this case, meiosis or some element of spermiogenesis, and even then the spermiogenesis would not necessarily be limiting. We can go into that later. We can actually get around most of the problems because of our platform.
Wow.
So thinking about this, which I do patient care. So if you have a male patient who has very poor sperm production. If you look at the literature, it shows that if you use testicular sperm, they'll do better than those few, meager sperm cells that the guy may squeeze out. But are you saying that we can take this man's stem cells, provide a better environment for maturation, and end up with healthier cells? And if so, you're looking at a much bigger population than the man who has no sperm.
That's exactly right. That's exactly what I'm saying. And
that is really exciting.
Yeah.
And biologically, it actually translates to the woman's side too, in terms of that egg quality piece. We can talk about that later, but it's nice to see the parallels between men and women, where the foundational cell, the starting cell, the cell that becomes the sperm, and then the egg cell is not the one that actually has the problem. It's everything around it.
It's the toxic environment.
It's the toxic environment.
Yeah.
That's a really good way of putting it, Amy.
So people talk about microplastics presenting in the testicle.
This is her big thing.
Man, I don't know how many times I've been asked about
Well,
microplastics
good.
the last 2 months.
I'm glad I'm not the only one. Is that one of the things that you think could be causing an issue?
Yeah, I mean, hypothetically, right? Like the problem with microplastics right now is that there's a lot of concern without a lot of evidence, right? So, I mean, even this these latest papers on finding microplastics in plaques in arteries, right? That maybe increase the risk for heart attack. There's actually no causal association, right? At the same time, right? When we talk about microplastics, when we talk about plastics in general, right? We think BPA, we think some of the other chemicals that leach out of plastics typically that are toxic. So is there a signal there? Potentially. Have we proven it? Not yet, But this is going back to my point about the environment impacting fertility, since that's our focus today.
Right.
I absolutely think that elements like this, like endocrine disruptors, like stuff that's in our water, stuff that's in the stuff that we touch in the built environment, like unless you're like living, I know, I'm going to die now. No, but all these things, maybe not all, but some of these things that we don't know about are definitely impacting our ability to
But that
have
makes
kids.
so much sense teleologically because, humans are supposed to reproduce. I mean, they're animals, right? And if we're having problems reproducing, instead of blaming the genetics, which everybody seems to, which would be the stem cell, workings, you're saying it's not. It's their progression through the testis from the base of the seminiferous tubule to the lumen of the seminiferous tubule that's dinging these cells.
Yeah.
So, and again, what is going on in that microenvironment? I think that's really, really exciting.
And I think, we've come closer to cracking the code than others because, and this makes sense, right? As we've developed this, it's made sense logically, right? What have people done to understand the genetics of infertility over time? They haven't been able to deconvolute this process because the technology wasn't there. What were we doing when we were at Baylor together? We were looking at we were looking at mutations in the rest of the body. We were never really focused on what's going on in the testicle because we thought that what was going on in the rest of the body was enough of a correlate to what's going on in the testicle. We actually know now that that's not necessarily the case, and that the reproductive organs are kind of doing their own thing in a way that creates both, genetic diversity, but can also create problems. But they also have their own processes, right? Like there was a paper that came out of the University of Utah last year that showed that men who are oligospermic, so have fewer than normal sperm, actually have more mutations forming in their sperm during the process of spermatogenesis than guys who have normal counts of sperm, right? And we never knew that until we actually started looking closer and closer.
So where do you draw the line, and where do you and your company sit in terms of optimizing genetics for disease prevention and for enhancement?
Yeah. So great question. So I'm going to start a little bit. I'm going to start just a little bit further upstream in the sense that, so there are two problems that I want to call out and address. And one is gene editing. The other is what is the source of the cell that you're using to get a gamete, right? And the reason I'm calling those two out is gene editing obviously is a hot topic. It is something that is, effectively banned, sort of kind of by the federal government. There are ways to still do it in the embryo, but it's becoming much more science fact and not science fiction, right? So from the standpoint of gene editing, look, we all need to be responsible, ethical stewards of the work that we do, right? There is great power in all of this type of work where, some might accuse us of playing God, right? And part of that is gene editing, right? What a lot of concerns around gene editing have been over the last decade or two have really been focused on off-target effects, right? So what happens if you manipulate a gamete genetically and you fix a problem using currently available systems, right? Largely that focuses on CRISPR-Cas9. This is really cool technology that lets you make even single nucleotide changes, right, in a sequence. But what's not known is where else across the hundreds of millions of bases in the genome another change can be made that can cause a problem that shows up in a human that's grown from this or that's birthed from this, right? That's been the concern, right? And until there was a lessening of regulation around like a total kibosh on making research embryos, right? And Korea has taken a big first step towards that. The American Society for Reproductive Medicine is now actually supporting that type of research and funding it fairly aggressively. They have a D&I grant. That's not a diversity. It's a development and innovation or something like that grant that supports that kind of research. We're not going to know the answer to those questions. So, to address the gene editing piece, I think there is absolutely something there. We need to be careful and thoughtful, deliberate, and very sequential in how we do the work. And we need to prove out that it's not going to, that fixing one problem is not going to cause a problem down the line. And there is a risk-benefit analysis there, right? Happy to go down that rabbit hole, but let me address one other thing. So what a lot of other people, scientists, at a high level, companies have been doing in the reproductive space in particular, and also in other cell-based therapies, they've been using a type of cell called an induced pluripotent stem cell, or iPSC. What that is in lay language is you take a cell from a different part of your body, skin, fat, muscle, whatever, and then you reprogram it. Like you push it backwards, right, to its more early state, and then you tell it to go down a different developmental pathway, right? Really cool science and very promising. But here's the problem, and here's where we need to watch out, right? Especially in the reproductive space. You can do that, and you can turn an iPSC, say, into a dopaminergic neuron for the treatment of Parkinson's, right? And then potentially repopulate the part of the brain that has that. And you probably will do okay, right? There's another project kind of looking at that for the formation of beta islet cells of the pancreas, which secrete insulin for the treatment of diabetes. In reproductive health, you got to be really careful. Why? Because that cell, if you push it to become a sperm or an egg, and I'm going to say that in heavy air quotes, there are still big fundamental differences between the genome of what a real sperm is or what a real egg is, and then what your iPSC-derived sperm and egg is, right? And the danger there is not so much in the genetic sequence, but in the epigenetic pattern, right? And what that is, epigenetics is essentially there are gates to allow genes to turn on that are mediated by epigenetics, let's just say, right? So those are the gatekeepers for gene expression. And if you don't create a pattern that looks just like a sperm or an egg, then you're not going to get the impact of a sperm or an egg. So until we figure out how to solve that problem, that is a very treacherous path to take in reproductive healthcare, right? And then the gene editing piece is it's another thorny thing, but it's something that we need to have on our horizon because you sort of said it, right? Like that is a key pathway to curing certain diseases that are otherwise impossible to cure. And so taking one more quick step back, and I'll let you guys jump in. When we think about quality control in our processes today, the goal is not to fix a problem. The goal is to weed out the cells that are obviously going to be problematic, right? And we do that with a panel of genes that we know that has been validated in the scientific literature to impact everything from spermatogenesis to fertilization, to embryo formation, to induction of pregnancy, to carrying that pregnancy to term, and then everything that we look for after a child is born, right? That newborn screen that you prick the kid's heel with and take a spot of blood, right? That's what we do now. And we're very clear that we're not going down the gene editing route until and unless we really know that there's an opportunity to do more good than harm.
So I guess on that same vein, in terms of the research necessary to warrant a certain therapy, what are you currently working on? Where are you in the progression of your research trials, and what's next?
Yeah, our next steps are to look at safety and preliminary efficacy. And, in terms of safety, we're doing some work in human studies in a petri dish, to sort of quantitatively show that what we're making is good, to show that we can definitively sort of weed out the good ones from the bad ones, and to show that they work. And then, we're moving into first in human studies, outside of the US, hopefully next year, that will show in human primarily safety and preliminary efficacy, right? And look, the goal here and our hypothesis, because of the quality control that we have, is that we'll be able to not just get higher IVF success rates, because when we talk about IVF success rates, it is literally how well the sperm fertilizes the egg and forms an embryo, but higher pregnancy rates. And if you look at IVF success rates now, right, IVF success rates in the best of hands can be maybe 60%, give or take, right? But that's just egg fertilization. Then that number ratchets down very quickly in terms of successful pregnancy and then successful live birth. Like you go 60, and then, you probably know the numbers better than me, but 20, 30 for induction of pregnancy, and then as a factor of age, like live birth can go down into single digits.
But what I'm hearing, and I may have misinterpreted this, is that your company may be able to help the oligospermic patient in that you are identifying those factors that the sperm require as they go from the stem cell to the mature sperm. Am I misunderstanding?
No, you're understanding it right. So, when we talk to our REI colleagues, the obvious starting point is men who don't have sperm in the ejaculate, right? That is the clearest population to help. But as you get into the quality discussion, you very quickly bring in severe oligospermics who have a low IVF success rate, and then go beyond that to exactly the types of guys that you're describing in your practice. So these are the guys who have failed at least one, if not usually multiple IVF cycles with their partner, and there's no clear female factor, right? Then it becomes a much more apparent case for a male factor. And look, it's going to take some time for the community to collectively prove out that there is an impact there. But we're at the point where we're going to start to do that in the next year or two.
So on a kind of similar topic there, going back to what you initially started Vault for, was to help men around the country, right? And so now there are all these companies, these telemedicine companies that are coming out to provide some of these services that you're describing. What do you think about some of these companies, and what do you think about the current need?
So I think the need is tremendous, right? And look, I mean, the need is tremendous because men are their own population, right? Men are different from women. Women have been banging the drum for decades, and they are very good at coming out and seeking help and making sure that they get it. Guys aren't like that.
Yeah.
Hence breast cancer success.
Absolutely. And when we think about in medicine, it's the guy who comes in, he's, 60 kilograms, he comes in with chest pain. So we think, a lot of medicine and most of the research is on men, but when it comes to male reproductive and sexual health, that's where men are kind of left out, despite the fact that there are so many medications to help men, right? And I have mixed feelings about this, because when you look at a lot of the people that are very loud online right now, who are incredible educators in like the menopause and the female medicine space, it's kind of this conversation of, well, men have it all and the women were left out. And I'm like, no.
So
that's
untrue.
not true.
Yeah, it's not true
Both populations of people are left out in this space.
Yeah. Right.
It's not a this versus that.
Right.
Yeah. And look, we very quickly learned at Vault that men are much more willing to have a phone call or a telehealth visit than they are to come into a clinic. Right. We also learned that men are much more willing to get care online than they are by physically going to a clinic. And there are a lot of frictions that go into that. So I think it's a huge unmet need. I think there is a ton of work to do. And look, the companies that are doing it right, and there are definitely a couple out there who I think are doing it right, should earn the right to win in that space. And as a result, help a lot of people.
But I have to call a timeout.
Okay, timeout.
I don't like telemedicine for fertility. I think it's great for low testosterone, hypogonadism, overall optimizing your health, getting your macros, all in line. But when it comes to fertility, the testing that you do is so significant, and we're not yet at a place where there are enough labs geographically to take care of men all over the United States.
So I fully agree with you on that, right? And look, there is...
And it's much more nuanced than writing testosterone prescriptions.
Agree. You have to see the guy at least once and examine him and make sure that you haven't missed something obvious. Right. Because those things don't show up in the diagnostics. I would say...
Like testis cancer.
Yeah. Great point, right? Yeah. How many of those do you find just by doing a male exam, right? Yeah. I don't know how many I found, But, at the same time, and also to your point, I think in the fertility space, we are in the dark ages of diagnostics, right?
Agreed.
I would love to hear your perspective on some of the new ones that are coming out, but bottom line is right now, sadly, I don't know, how long has the semen analysis been around?
1900.
Okay. So for the last 125 years, we have relied. It's true. Case in point, for the last 125 years, we've relied on a blood test for hormone, and that blood test didn't start till probably at least in the 1950s, 60s, 70s, looking at brain to balls hormones and a semen analysis.
He's such a scientist. Brains to balls.
Brain to balls hormones and a semen analysis to be the foundation of how we treat these guys.
Yeah, agree, 110%.
So do you worry with a lot of these online platforms that are making testosterone way more available to men that our infertility rates are going to go up?
Well, in the moment, sure, yes. There's... at the same time, I see plenty of guys who walk in from their, who have been cared for by their PCP and are on 200 milligrams of cypionate a week, and come to me and said, my doc never told me that this was going to make me infertile. Right. So yes, but we also know that we can recover the majority of those guys.
So I feel like for a lot of these online clinics and telemedicine platforms, infertility has to be a part of that. There's got to be this referral pattern where if someone comes in and they have certain levels and they check off that they are interested in current or future fertility, that needs to be maybe a referral to someone else who can have a more nuanced approach.
But what does that require? It requires a smart person on the other end of the telephone, i.e. the doctor. Well, and just because you're a doctor does not mean you know what you're talking about. Right. And that's one of the big problems we have. It's so easy to make money online with a telemedicine service, and yet is it really good medicine? On how much are you missing, and should everybody be able to do it? I just worry about poor care.
And that's where, that's where the nuance is really important and the platform is really important, right? Like, I mean, when I look at some of the companies out there, right, the fact that Peter Stahl, who's one of our colleagues in the space, we've known him for a long time, he used to be at Columbia, is now SVP of urology at HIMSS, is actually encouraging, right? And I say this because it's the standardization piece that's really important in making sure that these boxes are checked, right? Like, which happens pretty well in academic healthcare centers, which happens well in our practices because we have taken the time to create that. But also at scale, if it happens in these companies, and this is why there are a couple and not a whole bunch that are doing this right, then that question gets answered. Mm-hmm. But if it doesn't, you're right, it's a miss, and it's a I don't want to say liability, but it's a problem for the patient. And then that falls on somebody else to fix.
Mm-hmm. I think the question is it better for someone to get half-ass medical care than no medical care? And
that's the question.
Yeah, because you can get something on telemedicine. Mm-hmm. It's better than zero, but you don't want to hurt anybody. Remember, do no harm.
Yeah. And
I worry about the doing harm part, but I have to weigh it against at least somebody's talking to this person who otherwise wouldn't talk to anybody. So, you have to decide somehow to strike a balance.
Mm-hmm. Yeah, I mean, philosophically, I'd rather see people getting some care than no care, because I've seen the downside of that.
Yes.
But it is a fine line.
It is. It's a problem.
Yeah. Alex, to end our podcast on a more positive note, what gets you excited in the morning when you wake up?
Oh, man. A lot. I mean, look, what gets me excited in the morning is seeing how quickly and effectively we can get what we're doing in front of the patient, right? And it is what that day, week, month has to hold in terms of who I need to talk to, because this isn't a science project anymore. This is science fact, and it is touching the world, right? Like I would argue that this is the sort of first real public introduction in a meaningful way of what we're doing. But what gets me up in the morning is the promise of this actually touching patients in as soon as possible.
I've got to tell you something about Alex.
Yeah.
Alex, I don't know whether Alex is the Renaissance man, because he is into so many different things. He loves good pens. He loves good wine, right?
And he loves good sperm.
And he likes good sperm. So
I love good
he
sperm.
is complete.
I've stupidly thought about a vanity plate that had a reference to this, but I'm just not going to do it.
No, don't do it. But thank you again. You were great, as Amy and I thought you would be, and we're really excited that you came and shared this for the first time with America.
Yeah, well, thank you. It's a privilege to spend time with you guys. Yeah. Really.
Okay, thank you.
It is so good to see Alex today. I mean, I can't believe I met him, like about 15 years ago when we were doing research, and to see how he is potentially going to completely change how we diagnose infertility and manage IVF patients is literally blowing my mind.
Yeah, I knew that he had succeeded in maturing a sperm from a stem cell. I did not really understand until today how they've made these new discoveries concerning the things that are important to stimulate the sperm all along the way from the beginning until they become a mature sperm, which is going to give us, hopefully, a way to better diagnose and treat men with all types of infertility.
And you had actually mentioned earlier on in this episode some of the challenges that you have in some of the discussions and management options with your patients who are coming in who have only a few sperm, and then their IVF cycle doesn't work, and then you're left being like, what do I do now?
Exactly.
I mean, this could change the management of that patient and their ability to have a child.
Well, I hope it will change it to the point where we don't have to get to IVF so often, if we can figure out why their sperm production is decreased and perhaps treat it.
Yeah, so I can't wait to see what his company comes up with next, and as they proceed with what their current technology is going after.
Right, and I think we may have to have him back to talk a little bit more about what they're doing with oocytes.
Yeah, absolutely. Can't forget about those eggs and those ovaries. All right, Larry, so what's today's final F word?
So the final F word for today is phenomenal. Right? It's our new phonetic F word. Phonetic F word. Phenomenal, because I think it describes what we heard today in terms of new science.
Yeah, I love it. He's really turning science fiction into science fact.
I like that.
Yeah, it's a good topic. All right, Larry, why don't you give us some foreplay for our next episode?
Okay, foreplay.
Foreplay.
Yeah, so we're going to next feature Jeff Abraham, CEO of Promescent and a leader in the field of topicals for treating premature ejaculation. And I know it's going to be not only entertaining, but educational.
Thank you for joining us today on Fertility and Other F Words.
And don't forget to like, subscribe, and leave a comment.
Until next time.